let me, oops, yeah. Okay, yeah, well, it's a pleasure to give this talk for this community. I was sorry. Let's see, yeah. I know this cannot really be informal, even though that's what I guess I would prefer. So if somebody has a question, you can just either shout and I will hear it, or put the question in the chat and then maybe Kate can pick it up because I cannot see the chat. And there's a significant, if not tremendous amount of overlap with what I talked about at the Synthetic Cell 2020 meeting in New Mexico. So, but maybe, you know, if you have questions still, if you were there and you have detailed questions that remain from then, just let me know. And what I will do, I will mostly talk about the cytoskeletal work that we do in my own group and not spend so much time or no time on our national and European initiative to building cells, because I did that before. But again, if you're interested in knowing more about it in the end, I'm happy to answer questions on that as well. And yeah, so this is the first page showing most importantly the people who are currently in my group. So we're in the Technical University of Delft since now 6.5 years. I moved from Amsterdam to Delft in 2014, and I'm in the department that's called the Bio-Nanoscience department, which is one of the 6 departments of our Faculty of Applied Sciences. So that's a combination of physics, applied physics, biology, chemistry. And then together with the Quantum Nanoscience department, we have this umbrella organization, the Kavli Institute for Nanoscience. So together with the Quantum Nanoscience department, we have a postdoc program. We have Kavli chairs, visiting professors that come to Delft. So if you're interested, you can check it out on our website. So Bio-Nanoscience is a department that started about 10 years ago now. I wasn't there yet, but it was just a core of 2 professors, Nienke Dekker and Kees Dekker, no relation between the 2, who convinced the university to really invest in this research field at the interface of cell biology, molecular biology, and physics, mathematics. So they had a group focusing on single molecule biophysics experiments of DNA, protein interactions, mostly at the time. And then the university decided to start this new department. So at the moment, we have 16 groups, 16 PIs with backgrounds in physics, chemistry, biology. And we're together working on, you know, questions about biology at the molecular to cellular scale. So it's quite a fundamental department. And we co-teach a program together with Erasmus Medical Center, which is only 20 minutes by bike from us. So since 2012, we have a program there that we call nanobiology, and we educate both bachelor and master students with a mixed curriculum, you know, physics, math on the one hand, and cell molecular biology on the other hand. So, and in the context of a medical school, which is what Erasmus University is. So that's all very nice. My own group is listed here. So there's PhD students, postdocs, and technicians shared with other groups, most of them. And then I put 4 names: Nuria, Pierre, Sophie, and Matthijs. These are all PhD postdocs that were formerly in my lab. And of course, there were many others that I should credit, but I specifically show pictures that they made in this talk. So that's why they are here. But all of them have moved on to new positions since then. So we're interested in the cytoskeleton since many years, and what I called it now building minimal cytoskeletal systems for synthetic cells, because that's where we're going in the future. But since many years, we've been interested in reconstituting what I call functional cytoskeletal systems, sort of as minimal systems that help us to understand eukaryotic cytoskeletal systems better as they operate in real cells. So that's what I will be talking about. Oh yeah, that always happens. Then I don't remember anymore how to pass the slides. Oh, here we go. So just a little bit of textbook background on the microtubule cytoskeleton. So this is a pictures taken by, I should say, almost all pictures of real cells are not taken by us, except the fission yeast. This is what we do ourselves, but are stolen from various people's websites or textbooks. So this particular picture is taken by Torsten Wittmann from UCSF, and it shows a very nice picture of flat cells sitting in a dish attached to a flat surface where the DNA is highlighted in magenta, and the microtubule cytoskeleton, which is what we focus on, shown in yellow. And this is a fluorescence microscopy picture of a fixed cell, and it highlights one of the things that the microtubule cytoskeleton is doing, which is to provide a network of highway systems, you could say, for molecular motors to walk on. So molecular motors pass material from the inside of the cell towards the periphery and vice versa, with the help of plus and minus end directed motor proteins that use microtubules as rails. This is a very static picture, but I'm sure you're very aware that the microtubule cytoskeleton, unlike our own skeleton, is a very dynamic structure, not a static structure at all. And you see that, for example, very nicely in this, again, textbook examples of what microtubule cytoskeleton is good for in cells. And this is to build a mitotic spindle that is responsible for taking apart chromosomes during cell division. So you see here 3 versions of the same process. One from Kochakov lab, which is again a cell in a dish, from the Julie Kenman lab, which is a single cell C. elegans embryo, first cell division in this large embryonic cell. And then on the left, I think, is from a microscopy manufacturer website showing the dynamic process, where you see very nicely that, you know, this highway system that is present in an interface cell completely reorganizes itself, gets focused into 2 poles, microtubule organizing centers called centrosome, and then together they are putting the chromosomes first in the middle of the plane during metaphase, and then pull literally microtubules apart during cell division. So we've been for many years interested in understanding the dynamic properties of individual microtubules, and specifically also the force generating properties of microtubules. So when they attach to chromosomes, they can exert pushing forces against the chromosomes, but also against the cell periphery. And then during anaphase, the disassembling microtubules can also generate pulling forces, literally, when chromosomes move apart. So we've done, and I'll show some examples, experiments to just study the biophysics of these assembly and force generating processes. And then we're interested in how these dynamic and force generating properties contribute to the spatial organization of microtubules in the confined space of cells. Oh, wait a second, because this is where, oh, sorry. So I was going to just zoom in on the dynamic properties a bit more. So this is no longer a microscopy movie, but it's taken from the inner life of the cell. So it's now an artist impression of what an individual microtubule would look like if you were able to look at it at this level of resolution in a dynamic experiment. Of course, we can take nowadays beautiful cryo-EM images of individual microtubules and take snapshots of what these tube-like structures look like when they grow and shrink. But of course, these movies we still have to imagine. And this is showing a lot of details of what we know about the assembly process. So individual microtubules growing by 13 protofilaments that make a hollow tube. There's actually GTP binding and then GTP hydrolysis associated with this assembly, which you do not see in this movie. But the hydrolysis of GTP in the tube that is forming actually allows these tubes to occasionally switch from a growing to a shrinking state. It's called dynamic instability, and it makes for the property that individual microtubules in the same sample or cell can be either seen growing or shrinking, and that the intrinsic process leads to this switching between the 2 states, which, even though it was discovered in the 80s by Tim Mitchison and Mark Kirshner, it's still not completely understood how nucleotide hydrolysis exactly is connected to this switching behavior. So showing again how dynamic these polymers are. So like I said, we're interested in how these dynamic force generating properties contribute to organizing the cells. And we are inspired by not only cell division, which you see again here, but also we're interested, for example, in how this skeleton organizes itself in polarizing cells. So that's another model system that provides us with inspiration for our experiments. So you see a fission yeast cell in interface on the right, and this cell is about 10 microns long, 4 microns in diameter, and it's growing by an actin-based process only at the cell tips. And to signal to the actin machinery, there's proteins delivered specifically to the cell ends. So this is what I call a polarized cell, because these cell end markers are not distributed homogeneously around the periphery of the cell, but they are specifically localized at the cell ends. And I'm interested, or we're interested, in how the microtubules help to set up this pattern, as I call it. And if you're interested in protein patterns in cells, you may know, for example, the MinCD system in E. coli, in bacterial cells, which is a reaction diffusion-based mechanism that sets up an oscillatory pattern that also marks the cell ends, and this is then helping the cell to define where the middle is for cell division. But this pattern, even though, of course, it's not oscillatory, but it's also not a pure reaction diffusion mechanism, it really relies on a linear transport, which is what you see in this movie. So individual microtubules bring these cell end markers literally to the cell end, and that's how these patterns are set up. Okay, and our contribution, as I said, is to build minimal versions of that, and that's what I will go through in this talk. So first, the spindles. So let me take you back a little bit in time and sort of explain over the years what kind of in vitro experiments we've developed to study first individual microtubules and then build minimal spindles. So on the top, you see each time a real cell example of the process we're reconstituting in vitro, and on the bottom you see the in vitro experiment. So the simplest property is just dynamic instability itself. So on the bottom you see a turf microscopy movie of just tubulin self-assembling from dimers in solution, and the nucleation site here is just a piece of stabilized microtubule stuck to the glass, and there's nothing else here in this except for buffer and the right temperature and everything, no other proteins. So this ability to switch between growing and shrinking states is intrinsic to the microtubules, and people have reconstituted this since many, many years. And you see an example here from our lab at the bottom. So you can study it in a tremendous amount of detail if you're interested, how the switching probability depends on all kinds of parameters, specifically also, of course, how this changes when you add known regulatory factors of microtubules, of which there are many in cells. And if you look at the top, you will see the same behavior also in cells where individual microtubules reach the cell end and then switch from growth to shrinkage. The other property we've been studying and reconstituting is the force generating probability. So many, many years ago, when I started my own group, the first experiment we did is done by Marcel here at the bottom. And we use microfabrication techniques to make artificial boundaries, so obstacles, in this case, glass walls. And then, I don't know if, wait a second, oh, that's better. And then using DIC microscopy, we could follow the growth of these filaments against the barrier. And then in this looping movie, you see the filament bend as it hits the wall. And then from the bending response, in combination with knowledge about the rigidity of these filaments, we could, in a very quantitative way, deduce the forces that are being generated by the growth of these filaments. And Maurits is a more recent student who actually repeated the same experiments for reasons that I will explain recently, and now using fluorescence techniques. And this bumping into a barrier, buckling, and then generating a force, you see literally also in living cells. So the fission yeast on top, if you look at individual filaments, they reach the cell ends. And when they do, they behave exactly as they do in the in vitro experiment, which is proving that these pushing forces, in this case, are actually relevant for keeping the cell nucleus positioned in the middle of the cell. You don't see the cell nucleus here, but in the middle, these anti-parallel microtubule bundles are actually associated with the nuclear membrane. So we can study in all kinds of detail dynamics and force generating properties. I'm not showing it, but you can also do experiments measuring the pulling forces, and for that we use optical tweezers, and then stick the microtubule not to the surface of the cover slip, but to a bead, and then put a connector molecule at the barrier such that when the filament shrinks, it holds on to the shrinking, to the barrier while pulling the bead towards this wall, and that with an optical tweezer allows you also to measure pulling forces and not only pushing forces. So then, like I said, we're eventually interested in understanding how this all contributes to cellular organization. So, and for many years, and still in a way, we've been obsessed with the mitotic spindle. So the first question we asked is, if you take half a spindle, just a centrosome, with dynamic microtubules radiating in all directions, and we put on the cortex, if you like, or the edge of these microfluidic droplets, a connector molecule, in this case cytoplasmic dynein, it will hang on to the filaments that arrive at the cortex, and then we could show that the distribution of pulling forces that results from this cortical interaction actually leads the centrosome to position itself quite robustly in the middle of these confined spaces. And we argue that this is similar to what you see happening in the C. elegans embryos, where the spindle is also positioned with respect to the boundary of the cell with the help of dynein molecular motors found at the cortex. I will not have time to go into detail, but or too much detail. Actually, if you look at the embryo indeed more carefully, you see that this spindle is in the middle of the egg, but it's not symmetrically positioned. And a lot of cell biology work, including some laser cutting experiments, suggests that this asymmetric position is due to a higher density of force generators at the cortex on the right side of this embryo versus the left side. And that's what this leads to, this asymmetric stable positioning. It's an experiment in vitro that we would still love to do, and I'll show maybe at the end sort of what we're trying to achieve to study this asymmetric positioning. Anyway, this is all mechanics and naked polymers, at least in vitro, but of course in cells these dynamic properties are under stiff regulation of other molecular factors. So if we want to really build functional cytoskeletal systems, we also need to have this molecular control. And on the top you see an example again in real cells. This is from Anna Marnova's lab, with whom we collaborate a lot. And you see here at the tip of these growing microtubules a protein called EB3, labeled with GFP. And it's one of those plus-end tracking molecules. It's actually a hub that sits at the growing end of microtubules. It recognizes the specific structure, or how do you say, both the nucleotide state and the structure of the growing tips, and recognizes this as a platform on which these proteins specifically bind. And this platform recruits molecules of all sorts, some that allow these filaments to connect to kinetochores, to the cell cortex, some that regulate the dynamics by making this dynamic instability happen more frequently. So that is what we can now also reconstitute, and that you see at the bottom. So this is now an ester positioning itself in the microfluidic droplet with this control molecule at the end. And then, of course, we would like to put this molecule that is controlling the dynamics of the filaments ideally under some external control where we can change it in time, such that we can switch between different kinds of behaviors of this system by turning on and off ideally these control molecules. So I'm taking a bit of time to explain all this, because it's sort of the repertoire of things we can do in our artificial cells that is sort of now building up to ask more questions about complexity. Oh, I should say that just as a side step maybe, when we add these control molecules that travel at the ends of microtubules, we're of course also interested how force generating properties against obstacles change due to the presence of these regulatory molecules. And this is the reason why recently Maurits went back to what we call our buckling experiments, so that we can really, with high resolution, and that's what you see in this movie, study how filaments reach a barrier and and follow, you know, how long does it take before they switch from growth to shrinkage when they are in contact with the barrier, and how is this correlated with the behavior of these control molecules at the tips. And again, I'm not showing in details, but this is also a way of indirectly to understanding in greater detail how this switching event is exactly triggered. And this is because this green comet that you see is giving you an indication of how many non-hydrolyzed nucleotides are still at the tip. So it's giving us an idea of the structural stability, how this responds in, when you put the thing in front of the barrier. Again, if there's questions, you just can shout, but maybe not. Oh, sorry. Yeah. So then, how to go from this, everything I've shown you so far, to something that, more spindle-like. So just a step back to explain you how we make our artificial cells or microfluidic droplets. So this is not very original. Lots of groups do this, but we use like many. Sorry, I should. Stop here for a second. Yeah, so in the end, we don't want to build maybe complete spindles and all the beauty that you see in this picture, but we want to at least have a double ester and eventually also put something in the middle that resembles a chromosome, so that we can step by step understand better the force balance of this structure. As I've shown, we can look at single esters positioning, but we're sort of in a longer term program to add complexity step by step. And to do that, and this is what I wanted to show, we use these microfluidic droplets. So like many other people, we push water or a buffer that contains the centrosomes and the tubulin and anything else that is needed inside this compartment into an oil phase. And then we watch how the system self-organizes. So we cannot, for the moment, trigger anything. You've maybe seen the work that Joachim Spaatz is doing, where in addition to making these droplets, he also injects different components at different time points. We are not doing that, at least not yet. We put everything from the beginning. And in the case of microtubule esters, this means you keep everything on ice, nothing grows until it's all enclosed in these droplets. We make many of them, then we pipette them out of a reservoir into a slide that allows us to do spinning disk confocal microscopy, and then we observe the behavior of the esters that spontaneously form. And we can do that under different conditions. So in the cartoon here, you see the experiment that I explained before. So we can put a connector molecule at the periphery. It's a dynein molecular motor that is biotinylated, stuck to biotinylated lipids, at least in the old experiments. And then initially put a single ester, and then we can do theoretical modeling, which shows that as long as the filaments are long enough to reach all sides in the periphery and all sides, then the centrosome will position itself in the middle if you have the motor proteins. If you do not have the motor proteins, then this is what happens. So there's no generation of pulling forces in that case. There's just long filaments that get longer and longer, eventually bend and buckle. And that eventually always leads to the centrosome being pushed against the periphery and not sitting in the middle. The only way you can convince this thing to sit in the middle is if you carefully tune the average length of the filaments compared to the size of the container. As soon as the filaments become longer than the size of the container, this is the end result. And then if you put a molecular motor that pulls on the filaments, you instead drive this centrosome robustly to the center. And there's a little bit of statistics shown on the top. In the droplets, you can also sometimes catch two centrosomes, and without motors, the same thing happens, except that on top of the pushing to the periphery, there's now also an interaction between the filaments that gives a bit of a repulsive force. And this is just purely due to steric interactions. We believe you can do some calculations there to estimate these forces. And then if you combine centering forces due to molecular motors at the periphery with these repulsive forces, you actually quite robustly get this bipolar organization where two centrosomes do not sit in the middle, not sit at the periphery either, but find a stable position at a moderate distance from each other. And again, there's a little bit of statistics shown, but again, as long as the filaments are long enough, this is almost always what you get, which is a bit surprising. It's quite robust. Only if the filaments are too short, that this doesn't work. So then you may ask, okay, in a real spindle, that's what you see, but we know very well that in real spindles, there's lots of other components that are essential for this force balance. So, and I've said this now since many years, we can now use this very minimal spindle where we really understand the force balance, because we can model the complete thing and then ask, if you put additional force generators, how does this force balance change? And we've done it for a few components, but still there's lots more that we could do. So this particular picture, for example, has, in addition to what I just explained, also anti-parallel cross-linker molecules that you find in real spindles. So in real spindles, there's not only centrosomes of chromosomes in the middle, but there's also microtubules that overlap in an anti-parallel fashion and are held together by either passive or active cross-linkers that can keep these filaments together. And here we've put a passive cross-linker that sort of stabilizes the structure that I already showed. So what's next? I said we've been focusing a bit on other things, but we would still in the future, have a to-do list that we would really be interested in this experiment. First of all, not only look at the static outcome, but also to have some way of trigger the system, either by turning molecular connections on and off, so that we can also look at the dynamic process of this self-organization behavior. We would be interested, as I said earlier, in understanding asymmetry. So the idea, and I will show you in a second, would be to get some, for example, light-induced control on the interaction between dynein and the periphery. And then further in the future, if you have molecular or temporal control, we could also maybe learn how to switch this system from a metaphase to an anaphase type of organization. Again, to find what are the molecular strategies that allow you to switch between these different functional outcomes of the self-organization process. Eventually, it would be nice to also have some cargo in the middle, and that's a project that we've started to work towards, let's say, and for that we need a minimal a particle, let's say, that can interact with microtubules coming from both sides. So we started several years ago a collaboration with Andrea Masacchio from Dortmund, who is an expert in the human kinetochore and can make really isolated, reconstituted, complete kinetochore complexes. But within our collaboration, we focused on a minimal complex. So the idea would be to have kinetochore proteins that sit all the way on the microtubule end of the kinetochore that can make a reliable connection between dynamic filaments and beads, for example. And what we've done so far is really figure out, together with a postdoc, Pim Huizenveld, in Andrea's group and Vladimir in my own group, we've shown that if you have a molecule called NDC80, which is at the microtubule end of these human kinetochore complexes, if you have just a single of a molecule like this, it cannot faithfully track a shrinking filament, for example, but we could do an experiment where we could count and show that if you have a complex of at least three of these NDC80 molecules, that gives enough reliability, and that's what you see in these green dots here, following shrinking filaments to reliably stay attached to a shrinking filament. And we put this also on beads and showed that it can also pull a bead with a certain amount of force through this experiment. So in the future, we would like to coat beads like this and put them also in our minimal spindles. The other technique we've been struggling with and getting some progress is this light-inducible connections that I talked about. So the idea would be to not only have spherical droplets, but also, let's say, C. elegans shaped droplets, and then put them in what I call parking spots in the microfluidic device, which keeps them in a certain shape, and then not put biotin strapped directly on the dynein, but have a light-inducible dimer system. We're now actually not working with the law of EPDZ anymore, but with SSPB eyelet. And that works actually very nicely on surfaces. So you can put one half of this dimer on the surface, then shine light, and it will recruit the other half, in this case, attached to just a fluorescent protein. But in droplets, that turned out to be a lot harder. So we now have sometimes, on good days, experiments where we can put the second component in the lumen of these droplets, shine light, and then recruit this to the periphery. And we want to do this with dynein and then shine light in an asymmetric way to be able to, to recruit force generators asymmetrically in these elongated droplets. Okay, let's see, I should maybe speak another 15 minutes or so to also leave some time for questions. So I want to maybe a bit more quickly show also what we are doing. It's a bit more work in progress on trying to reconstitute a minimal system for cell polarity. And like I said, I'm just this question of how can you use linear transport by a filament to set up a reaction pattern, what in some other systems is done by simple reaction diffusion, is something that sort of fascinates me. And just to explain what we're doing, I have to go back to the cell biology a little bit of this problem. So what I'm showing here is on the bottom, two real fission yeast movies. On the movie is where we again see this T1 molecule, it's the cell end marker being transported to the cell end. And this is downstream of this T1 molecule is then signaling molecules that recruit the actin-based machinery that is responsible for cell end growth. But what's also interesting is that derived from this T1 pattern, there's other patterns like the POM1 that you see here is a more diffuse signal, which is again localized at the cell ends, which like MinCD is responsible for recruiting the cytokinetic ring to the middle of the cell as soon as the cells are long enough. So this whole system of cell end markers is very complex. I put a cartoon with, I don't know, these are 8 molecules known to be involved in this whole system, but there's maybe more. And there's feedback between all of them. So it's very difficult to figure out exactly what is minimally required to set up such a pattern. Also because the same pattern is actually used for biologically distinct functions like actin recruitment or measuring how long the cell length cell is in the case of POM1. So before we did anything in vitro, we asked, okay, you know, since microtubule tips are already pointing to where the cell ends are, you may think that none of this complicated machinery is necessary to provide a basic cell end pattern, because you know, we reasoned if you have a molecule that diffuses in the membrane freely and you allow this molecule to interact with the plus ends of microtubules, then simply because of the fact that the cell shape is what it is, and therefore cell end microtubule ends contact the cell ends more often than that they contact anywhere else the membrane. And if you then also assume that these diffusing molecules can stay bound to this microtubule ends for some amount of time, you may bias their presence at the cell ends without doing anything else. And this is in fact the case. So we did an in vivo experiment where we made a completely artificial chimera protein, which is, has a membrane anchor. So if you disrupt all the microtubules, this protein will be, or this chimera will be homogeneously distributed everywhere in the cell. And then if you, I have to, oops, 1 second. Okay, no. So as soon as you allow microtubules to grow, then indeed you bias the presence of this molecule to the cell ends simply because they reside there longer than elsewhere through the interaction with microtubules. So this is what I would say the basic pattern that you can get, and it's only based on microtubule protein contact. So of course, if you look at the dynamic properties of this pattern, it's very different from the real one, because when the microtubules are gone, immediately the pattern is also gone, and this is not true for the biologically functional patterns. But still, to know if this is the simplest way that you can set up such a pattern, the first thing we wanted to do is reproduce this in vitro, because this looks straightforward, and we made the chimera, it's artificial, and we can prove that it's primarily at the cell ends. But since you never know what other proteins are secretly contributing to this process anyway, we wanted to also demonstrate this mechanism as a first step in vitro, and this is what Kim did. So this is now very much I would say, an engineering experiments where we sort of put together all kinds of bits and pieces to make this experiment work. So what she did is make microfluidic droplets with microtubule esters. So it's not an elongated shape or anything yet. We just wanted to prove this principle that if you have microtubules in contact with a cortex and some affinity between the molecules and the microtubule ends, that you can create hotspots of membrane proteins that way. And to show that, she used microtubule esters with end-tracking molecules that had an affinity for a peptide that we put on the periphery. And the components that you see here are, we got them from all kinds of different sources. So this demonstrates how, we rely a lot on collaborations and are very grateful to help from all sides when it comes to biochemistry. So the artificial centrosomes she used were actually beads, that nucleated microtubules that we got from Tim Mitchelson's lab. And we use nickel and TA lipids that you can just buy, and then an end-binding protein that is fluorescently labeled from Michel Steinmetz' group, and a his-tagged peptide that recognizes these end-binding proteins from Anna Manova's lab. And an additional technical addition here is that to see microtubules clearly in these three-dimensional droplets, we wanted to highlight the microtubules better. So we use a microtubule-associated protein, MAP7, that has a nanomolar affinity for microtubules, so that we can see, even though there's a background of tubulin here, that makes it normally hard to see individual microtubules, makes it a bit easier. So if you do the control without any microtubules, you will see that the end tracker is just in the lumen of these droplets. It's the green picture. The cortical peptide is at the periphery. This is the yellow picture, and it's homogeneously distributed. And in the absence of any microtubules, the MAP7 is also homogeneous in the lumen. Now, if you add centrosomes and microtubules, this is what you see. And it's now just a single plane in 3D, and we're going up and down with the spinning this microscope. The microtubule ester is not very big, so it's actually a couple of beads stuck together, and then individual microtubules in red reach the periphery, and whenever they hit the periphery, you see a yellow hotspot appearing, and we interpret this as an end interacting with these cortical peptides, which for a short amount of time recruits a hotspot of these otherwise diffusing molecules, like we were suggesting in the efficient yeast. So this is very exciting to us, that we can create hotspots with the help of dynamic microtubule tips. And just to convince you that if you do not put any nickel and TA lipids, so I have to say, I didn't say it, I think yet, that the peptides we use is his-tagged. That's how we recruit it to the cortex. If you leave out the nickel and TA lipids, then this cortical peptide goes in solution, and you get normal comets just traveling at the ends of microtubules, and nothing is now stuck at the cortex. Now, if you go from this experiment, add nickel and TA lipids, you now introduce a connection between these yellow proteins and the cortex, and you go to the experiment I just showed. And what we wanted also is to quantify how long do these contact points last? Do they have a stabilizing effect on microtubule dynamics? Can we really demonstrate that these spots only happen where the microtubule is hitting? So in the next movie, what Kim did is to take all these 3D slices and project them onto one plane, and then you can really follow these hotspots in time and space, even though they go up and down and Z, and analyze how long these contacts last. And maybe for the microtubule community, interesting detail is that if you take a peptide that is interacting with the microtubule end, but also slightly with the lattice, you get a completely different behavior that you see on the right. As soon as microtubules hit this cortex, the lattice starts to be recruited to the periphery because of this sideways contact, and the behavior of the ester is completely different. It actually, as a spider, gets sort of pulled towards the periphery. And I said this is interesting for the microtubule community because there's actually different cortical proteins that have these different types of interactions with microtubule ends, and also in cells lead to different kind of behavior. So we have a model system to study that different. Okay, anyway, we think we have a proof of principle that these hotspots can help organize the membrane. And what we now, of course, want to do next, and that's what we're in the middle of, is to go to elongated droplets to see if we can get cell ends or droplet ends to be highlighted sort of as we see in cells. For these elongated droplets, we needed to go away from centrosomes and to seeds, and this is just dirty details, but microtubule seeds have a much harder time nucleating microtubules in droplets than they do outside, for reasons we do not understand, but the trick is to put a little bit of crowding agent and then it nucleates again. And then we want to, of course, combine this elongated shape with the cortical recruitment, and that's what we're in the middle of doing. I want to maybe, if I can, Kate, show 2 more slides of an additional element to these experiments. And that is, if I go back to this introduction, I said that the pattern that we have with this chimera protein that we're now in the process of reconstituting also in droplets is very transient. So as soon as the microtubules are gone, also the pattern is gone. And one of the reasons, or one of the ways to make a more lasting pattern would be to not bring individual molecules, but actually bring clusters of molecules that accumulate at cell tips. And this is what seems to be the case in real cells, so that these cell end markers are not just interacting with the microtubule tips, but in response to being at the tip, they actually seem to make multivalent clusters. So in separate experiments, we've shown that indeed, and this is now not with droplets, but with microfabricated chambers, that if you take a functionalized wall and you bring not individual molecules, but clusters, we call it. So these are molecular complexes that make multivalent particles, that individual microtubules, if you look carefully, can bring these clusters to a functionalized wall, and then they will stick there and remain even when the microtubule disappears. So we think this might be a mechanism for creating longer lasting patterns. But this is something that, like I said, we want to in the future combine this particle transport with cortical interactions and elongated shapes to see if we can not only get patterns, but also patterns that last a bit longer than just the contact time of an individual microtubule. Okay, I think I've talked a lot. I want to maybe just go back a little bit to this larger project. So in my title, it said, building cytoskeletal systems for synthetic cells. Of course, in the past, we've really been motivated by understanding the mammalian cytoskeletal, or the eukaryotic cytoskeleton, I should say, not just mammalian, but the eukaryotic cytoskeleton in a bit more complexity than probably would be needed for synthetic cells. So we're now thinking how to use this same technology, but take a step back and go to simpler systems, because for this synthetic cell that we are doing together with you and other people in the Netherlands and elsewhere, we need to worry not only about spatial organization and cytoskeletal structures, but lots of other things. So we need a simpler system that we combine with these other modules, and it's even feasible that to build a minimal synthetic cell, that a complex cytoskeleton is not needed at all. We will see. But to make a step in this direction, what we're doing is to study a bacterial analog of the eukaryotic microtubule. So one of the things we would like to do, together with Christophe Danelon, our colleague in Delft, is to also put these cytoskeletal polymers under the control of a gene expression system, where we can control gene expression of cytoskeletal elements that then shape or organize the cell or not. And this is impossible with eukaryotic tubulin. It simply cannot be expressed in the pure system or in bacterial extract. So we're switching for this purpose to a bacterial version of microtubules. There's a few publications several years ago. And one of the students in the lab is actually repeating these old biophysical experiments with now 4 filament microtubules instead of 13, and also measuring the forces, studying the dynamic behavior. And then together with Christophe, we would like to put this in liposomes under control of a gene expression system, and maybe, for example, either use these filaments to make patterns, to transport DNA, or to even change the shape of these liposomes. So that's just a single slide. I think I will stop. I hope gave you the impression of the kind of experiments we're doing and would still like to do in the future. And I'm going to skip this one and thank the many people we collaborate with over the years, the funding sources, and most importantly, my group. And I always say it, if you've never been to the Netherlands and you like walking on the bottom of the sea, then maybe not now, and certainly not at the distance that you see in this picture, but sometime in the future, we'll be happy to take you on a trip like this. Thanks.
Thank you so much, Marileen. That was amazing. As always, your pictures make me jealous. I guess I would like to start. If someone has questions, you can put it in chat, but I would like to start with kind of a more of a general question, which is when you're putting those structures into liposomes, and presumably you want to end up expressing it from genes.
Yeah.
In situ, how would you control the expression rates and ratios of different proteins?
Yeah, well, you know, that's a good question.
That's what we all struggle with, so I hope you figured it out.
Yeah. So you would have to ask Christoph. So for the moment, we're just in a phase where we see if expression of these systems at all works, and if they then form filaments. So I'm not showing the pictures, but let me see. Was it? No, I didn't put it in here, but together with Christoph, we've already shown that this bacterial tubulin that we use, BTAP A and B, it can be expressed by the pure system inside liposomes to the extent that filaments will form and also deform the vesicle, make these elongated vesicles. So this can be done, and now you want to have either control over the expression level of the tubulin, but I would be more interested in the control of some regulatory molecule like we have with the microtubules, where you can switch from highly dynamic to more stable filaments and make them very short or long. That's how the cell does it, not by expressing more cytoskeletal protein, but by controlling the regulatory molecules. So for the, and there, Christoph and also others work on clocks, on oscillating gene expression networks. So I think it's still a way to go, but if you find a way to put a regulatory molecule under temporal control of expression, then you can think of doing these things. For the moment, we have plenty of examples of molecules that we can express that control eukaryotic microtubules, but we haven't found candidates yet for the bacterial microtubules. So they work fine in the pure system, but for now we don't have a candidate yet for controlling their dynamics. But we're trying, you know, with mass spec experiments to find binding partners to do this. Yeah.
Yeah, that sounds like, at least to me, that sounds like that would be the hardest. So you have a couple of questions in chat. Can you see?
Yeah. From Ankit, I remember you said that the microtubules buckle when they encounter the edge of the membrane. Is this buckling effect responsible for concentrating the number of microtubular tips at the cell poles in the non-spherical cells? I think partly yes. So what I've shown is microfluidic droplets that are like hard containers, but as soon as you do this in a liposome, which is very deformable, then for sure in a liposome, when you have multiple microtubules, their tips will focus and they make a bundle, which then actually deforms the membrane quite easily. And I think in migrating cells or neuronal cells, a similar effect can happen, you know, where shapes or for example, Philopodia even can really concentrate microtubule tips and bundle them. So this is definitely what you would expect and what you see happening in the liposomes very easily. I don't know if that answers the question. Then there's Alex. Fantastic work, very interesting, thank you. My question is related to your work on non-spherical vesicles. In your opinion, what's the main challenge towards inducing shape change in using these bottom-up cytoskeletal protein-based approaches? Is it an issue of concentration? Not using microfluidics, but a bottom-up approach. Yeah, so I think I maybe to some extent I explained, so if you express with the pure system bacterial microtubules, they will deform liposomes. Many years ago, people have also already included eukaryotic tubulin with different methods to making liposomes, for example, these electroformation methods or swelling methods. And if you then put in a eukaryotic tubulin and you raise the temperature to a temperature where the filament starts forming, it will also deform the vesicles. So I think as soon as you find a way of encapsulating these proteins inside the liposome, there's no way around that they will deform the liposomes. Typically, a cell is much harder or much stiffer, a cellular cortex, than a simple liposome, which is maybe too deformable to be a good model for any cell. Ali asks, I didn't catch this point on how you can control the elongation decomposition rate of microtubules in your in vitro system. And so I went maybe a bit fast over it, but in the beginning, these end-binding proteins that travel specifically with the microtubule ends, many of them have a strong effect on what we call the catastrophe rate. So it's the probability to switch from a growing to a shrinking phase. So if you have none of these molecules around, microtubules tend to grow very long and only switch occasionally, but then these different associated factors do different things, but there's quite a few, including this EB3, which enhances the probability to switch, and as a result, microtubules on average remain much shorter and more dynamic. And then there's other proteins that really, like MCAC, which will also decompose microtubules. So there's a whole zoo of proteins that have any effect you may desire on the dynamic properties of microtubules that you can add to these systems. Actually, we tried also in the pure system with Christoph, we could encapsulate eukaryotic microtubules in liposomes with the pure system. We could not express the eukaryotic tubulin simply because it doesn't have the required chaperones. It's anyway a protein that you have to isolate from tissue. You cannot express it in E. coli, for example. It doesn't fold properly, but we could encapsulate it, and then in the presence of an active pure system, and we then had one of these control molecules expressed by the pure system, and that we could show that when this protein control molecule was expressed, the microtubule became much shorter than when it was not expressed. So that's sort of a first step towards control.
Thank you very much. That was really awesome, Marileen. If there are any other questions?
This is not a question, but just a statement. Need a combination from Alex again. Need a combination of membrane properties and shape-inducing proteins. Yes, or an actin's cortex, for example, which gives mechanical rigidity or mechanical control to the periphery. So you would need that. Yes.
Okay, thank you very much. If there are no more questions or comments, we will post a recording of this seminar, and thank you so much for taking the time, Marileen.
Yeah, it's a pleasure.
And see you all.
Now my evening starts.
And my day starts.
Yeah.
See you all next week.
Yeah, do you know who's speaking next week already? Or?
Yes, let me pull up the schedule. I should know, but it's going to be Ayush Padney from Caltech.
Ah, okay. Okay, well, hope to see you soon for real at some point.
One day we'll travel again.
Yeah, one day, yeah. Okay.
Okay, bye everyone.
Bye.